Today is not a difficult class, but I do recommend you to pay attention because it is a very important class. It's not that difficult if you are with me from the beginning. So what are we going to do today is, in fact, we're going to do the process of transcription and translation, like you can see behind me. So transcription is DNA into RNA, and this happens in the nucleus. You know that because the DNA cannot leave the nucleus. And then translation is RNA into protein, and you also know that happens. Who can tell me where that happens? Translation.
Ribosomes?
Yeah, in the ribosomes, yes. The ribosomes, they do the translation, but where is it happening exactly?
Well, they can either be on the ER, the rough ER, or in the cytoplasm sometimes. It depends.
Really correct, Anna Sophia. It pleasures me to see that you give me a double correct answer, because it's exactly that. Okay, so let's have a look to the presentation of today. So we're going to see those two processes, transcription and translation. And then I'm going to introduce a little bit about biotechnology. Okay, we don't have the time to see so much about it, like I told you yesterday, which is a pity because it's very interesting. But anyway, we can touch certain parts, and tomorrow we will do an activity about it. And also tomorrow we will do a quiz about DNA and about what we see today. Okay, because don't forget that next week, Thursday, you already have your second partial. So I need some quizzes as well in order for you to have your grades. Okay. Let's have a look at the presentation, okay? I'm not going to explain all the text. And again, I'm going to ask you, don't be afraid of this. It's really not that hard, okay? I try to go as slow and as calm as possible and explain everything. So let's go back to yesterday. First, we go back to yesterday, and what you see here is the DNA strand, okay? We have seen the DNA strand, and we know that DNA has to be copied, okay? The DNA has to be copied because before a cell wants to split from one cell into two cells, which we're going to see next week, the DNA has to be copied because each cell needs a whole set of DNA. And this is what we call DNA replication, and this is what we have seen yesterday. Okay, let's do a little exercise about this to see if you understood this. I think you did, but we can quickly see that. With a pizarra, it's nice to do this. There we go. So I'm gonna put here a DNA strand. I'm gonna invent a DNA strand. It's completely random, okay? So if I have a DNA strand, what will, if it starts with A, what will be the complementary thing? So let's
T.
Okay, that's T indeed. So let's make a DNA strand. A, T. T. If I have G, it will be
C.
If I have C, it will be G. If I have T, it will be
A.
Yes, and that's how we go on. I just built something. So okay. So that's our DNA strand. When we do DNA replication, okay, the DNA strand gets copied. Okay, let's do that also. Okay, DNA replication. Let's replicate this DNA. So please help me, because I forgot how it replicates.
AU.
If I have a T, A, it will combine with a
U.
T. No, we're not RNA.
Okay.
And the T will combine with a
A.
Okay, well done. The G will combine with a
C.
and the C will combine with like this, okay? Everybody understands what I'm doing? This is wrong.
Yes.
Okay, let me
Yeah.
finish quickly. Okay, so this is what we have done yesterday, okay? I'm gonna take it in line. So what do we have basically is here, we have here this strand of DNA, okay? That's this one, okay? And then we have the other one, which is here on this side, okay? It's this one, okay? And you know that the DNA opens and then we fill it with the new DNA. Let's put that in blue. So what we have here, this is the newly formed DNA on the inside, okay? And this in the end, this goes to one cell and this one goes to the other cell, but in the very end, they're identical, okay? What we're going to see today, so this is DNA replication. This is separate from the rest. What we're going to see now is basically transcription, okay? We continue with that in a moment. Transcription, okay? So what is transcription? Transcription is DNA to RNA, okay? If we take the same strand of DNA, how will it be translated?
AT or how?
So
AU,
let's see that
since
in the
it's RNA.
presentation. I come back to this in a second. Okay, so this is the DNA replication. You all understand that. And now we go to the transcription, okay? So transcription, dear students, this is happening in the nucleus, okay? Because you cannot take the DNA outside the nucleus, okay? So what do we see here is a DNA strand, okay? And here the transcription is happening in the nucleus, and this is the messenger RNA that I get, okay? There's something interesting you have to understand. You see that this RNA is almost identical than the top one of the DNA. So basically, if this is a gene, this is what you want to copy, okay? And this was what we call the coding strand or the non-template strand, like I sent you in the message of WhatsApp, okay? And the bottom one is the template strand. The template strand is giving us the code. So basically, if this is a gene from A to C, and this is the gene you want, you have to copy the template strand because otherwise you get the opposite. Okay, everybody gets that?
Yes.
Yes? Okay, so let's have a look in the Pizarra how that happens. So, let's say that the green from T to T is my gene, okay? Let's suppose that the green thing is the gene. Green strand is the gene. Okay, we call that where the gene is, this is the coding strand, okay, because this has the code. So in fact, if you want this gene in messenger RNA, what you have to do is copy the template strand, which is the red one. Okay, let's do that. One student told me yesterday already that there is a difference between DNA and RNA. One difference is that DNA is double-stranded, RNA is single-stranded, and DNA stands for deoxynucleic acid and deoxyribulose nucleic acid, and RNA stands for ribulose nucleic acid. So it's just a chemical difference. And then the other difference is that DNA has the thymine, but the RNA doesn't have thymine, it has uracil, okay? Which we see in a second in more detail. I just want you to tell me what will the messenger RNA be if the green one is the gene we want to copy. So if this is the gene you want to have in the messenger RNA, you have to copy the template strand. So how does that look like? A combines with what?
U.
With you. With you.
Because T doesn't exist. Then I'm going to do it in red because no, in green is fine. Green is fine. U. Then G will be?
C.
C will be?
G. G.
G.
G will be?
A.
No, U. Yeah,
The T?
T will be A, another T, G will be C, C will be G, and A will be
U.
U.
Very good. That's transcription, okay? It just copies one side of the DNA into RNA with the only difference that a T becomes a U, okay? So let's go back quickly to the presentation to see the details about this, okay? Okay, here the DNA becomes RNA. Where is it? Oh, okay. So here, DNA double strand, RNA single strand, DNA deoxyribose, RNA ribose, okay? The nitrogenous bases are ATCG, and in RNA it's AU and CG, okay? The kinds, there's only one kind of DNA and there's 3 types of RNA, which we're gonna see in a moment. So let's go, now we have understood transcription. Let's go now to translation. Translation is the messenger RNA we just made. We're going to translate that in a protein, okay? So I go back to my pizarra in a moment, but before we start, there is something you have to know. I explained yesterday that DNA and messenger RNA use a code, okay, with letters A, G, C, in RNA it's U. And in fact, every 3 bases is 1 codon. And every codon codifies for 1 amino acid. You know that amino acids are the pearls of the string making the protein. You know, a protein is a string of amino acids and then they form a protein. And this is how that works, guys. Look, a codon has 3 bases. The first one, the second one, and the third one, okay? The first one can be 1 of 4. The second one can be 1 of 4, and the third one can be 1 of 4, okay? So now I'm gonna switch all the time because I want you to translate our messenger RNA in a protein, okay? The ones that we randomly made in the pizarra. So I go back to the pizarra quickly because here we have our messenger RNA. And the first codon is UCG, okay? So this will become 1 amino acid, okay? Because a codon always is 3. So let's go to the presentation now and see, I'm gonna write them down, otherwise I have to switch too much and I lost a lot of time in the other group by doing this. So I have UCG, I have AAC, yeah, and we have G and U, that's not enough. So we have 2 codons here, not 3. So we can have 2 amino acids with this little piece of RNA. And we're gonna have a look which amino acids they form, okay? And to do that, we need that little table. You don't have to know this by heart, guys. It's just for you to know how it works, okay? So our first codon is U. The first base is U. The second one is C. So I'm here in the U and in the C column. And then I have to select which one of the four it is by looking at my third base, which is the G, which is the green one. So I have UCG. Which amino acid is it?
Serine.
Serine. Yeah, very good. So all these names here, all of these are names of amino acids, okay? And then you see here stop and start. These are special codons in the DNA that tell the ribosome to stop. If you have UAA, UGA, or UAG, it means it's finished, the protein. And almost every gene starts with AUG because this is the start codon. It means start to produce amino acid. Let's have a look to our second codon, okay, which was AAC. I have A here, I have A here. So I'm somewhere here, okay? And then I have to look at my third base, which is C, which is, what is my amino acid here?
Aspirin, or I don't know how to say it.
Asparagine. I don't even know how you say that in English, asparagine or asparagine. I always used to say asparagine because it's more Flemish, but I really don't know what is the correct pronunciation, but it's not that important. So let's go back to the Pizarra. And here you see, I'm going to write it down. What are our two amino acids? This was serine, okay, and this was asparagine. Okay, and then in the end, in fact, what you get by connecting all these kind of amino acids together, you get like a chain of amino acids, and this is causing the protein to form, okay? So you get another one here, you get another, it will be connected to another one, and then in the end, what you get, it's really long. I just give two examples. What you get is like a really long protein which folds and looks like that. This could be a protein, or one other protein that you all know, it looks more like a Y, which is an antibody, okay? So that's basically how transcription works. We still have to see certain details, guys. So wait a second before ending this. So here, the codons, you understand? So this is, let me go back here. We had DNA to messenger RNA. This is called transcription. Then we have the messenger RNA into the peptide, which we call translation. This is happening in the ribosome near the endoplasmatic reticulum or the cytosol, like, who said it? Anna Paula, or what was your name? Yeah. Ana Sofia, sorry. And each codon consists out of 3 bases, and each of those combinations represents a different amino acid. Okay, clear so far? Let's see some.
Kind of.
The DNA here, the DNA goes open. You have here the gene you want to copy. This is the gene you want, but the RNA will grow on the other side to make an exact copy of this one, okay? So that is transcription. Okay. And then I have to tell you something else, okay? Because in fact, in the process of translation, there is different kind of proteins, okay? So here you can see our messenger RNA, okay? And the ribosome is what we call rRNA, or ribosomal RNA, okay? So this is the rRNA, okay? But like I told in the other group, the ribosome reads the messenger RNA, it has the codon, and it brings in fact, an amino acid, okay? Because the amino acid you need to make the protein. And what is bringing the amino acid is this thing, what you see here, is like the green thing. And this green thing that brings in the amino acid, this is what we call the tRNA, or the transport RNA. So let me explain that here with this image. You see here the messenger RNA. Here you see the ribosomal RNA. Here you see the peptides, or the amino acids that are growing a protein, okay? And this one will move forward okay? And when it moves forward, then it will be coded by a messenger, by a transport RNA, and this one will bring the correct amino acid, okay? So we have a lot of transport RNAs that select for each codon the right amino acid, and they bring it here, they sit in front of the other one, and then the protein connects to the new amino acids. And you see here how it's growing longer and longer. And this is thanks to the ribosome does that, in fact. So the ribosome here, it's reading the chain, but who is bringing, in fact, the material to build the protein is this tRNA, okay? And here you see how the chain grows and grows, okay? So every time there is a new one coming in front and the chain becomes longer, okay? I show you that on a different image. Here you have the DNA, okay? So here you have transcription, okay? Here's the messenger RNA. The messenger RNA is here in the nucleus. It travels outside the nucleus by the nuclear pore, and here you have the messenger RNA. Then the messenger RNA combines with a ribosome, okay, like you see here. And the ribosome, which is the ribosomal RNA, it starts to read the messenger RNA, okay? And here you see how that happens. The chain is already forming because it's reading the codons. The last codon is thrown away, but in front, the transport RNA brings the correct amino acid for the next codon, and then the protein grows, and it comes longer, 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 longer until the protein is finished. Okay? Is this clear, guys? This is how this transfer RNA looks like, transfer or transport RNA. So here you see it's a molecule. It has a special anticodon which reads, in fact, the messenger RNA, and it's the ribosome putting everything together, and it brings the correct amino acid.
I have a question.
Yes, go ahead.
Like, what codons are thrown away? Like the one in the picture. Like, is there something specific to know, like which ones are thrown away, or it's just randomly?
What do you mean thrown away? I don't understand.
In a picture, I think it's
This one?
lower. No, the other one.
This one?
The pink one.
This one?
Like, yeah. That one. The
Yeah.
one that says CCC.
Ah, no. In fact, they're all thrown away. So basically what happens, so there's 3 positions in the ribosome, okay? In the middle position, you have the protein that is already formed. In the first position is the next codon that will be connected to the protein, and the ribosome moves, okay? So this one, in fact, moves to the front, and this one becomes second position when it glues, and then a new one comes to the front, and the last one without anything anymore, because this one will move here, it will be thrown away. So every transport RNA, every transfer RNA is thrown away. I don't know if I explained myself. It's like a little train, in fact. So the ribosome is reading, okay? Maybe I can draw on top of this. I can try.
Like they are switching places.
Yeah, they're switching places. So the yellow thing is moving forward, okay? And every time it moves forward, a new transfer RNA will sit in front, okay? And then the chain will switch to the next one, and the empty one will be thrown away, and then you have a new one, and that's how it grows. Maybe we should see it in a video. There is a video about it, which I can show you. It's in, maybe if we have time to see it, we can see it. Let me quickly go to the Canvas content. Maybe there is a video that explains it. I don't think we have the time to see it, but let me go there with the Canvas content to go over it, everything again with different pictures that you really understand. So wait a second, let me open it. I closed it by accident because normally I always have everything open before the lesson starts. It's really not that hard, guys. It's a very simple process. If you want to know all the details, it becomes very complex, but we just see the basics of this genetics, because in fact, genetics is a whole science on its own, you know. I mean, I got genetics in university and it's like a book of 1,000 pages if you want to know everything. So it's very complex. But it's very, very interesting. I think it's amazing how they all discovered these kind of things. It's very intriguing. It's very magical to see how they all found things out and how complex life is, in fact. And don't worry also, because we will come back to this in the future. There will be more about DNA still. Okay, it's finally open, so I'm going to share screen. And we go over it all again so that everything is clear, because I want it to be clear for you. Okay, so this is what you have in Canvas. You also have the presentations, okay? We have an index here. Some people, they don't know the difference between genomics and genetics. It's very similar, in fact, with the big difference that genomics is the study of the genome. So the genome is all the DNA, the complete DNA of a certain organism. And genetics, it's more the study of a piece of the DNA. So a particular gene, okay? Could be antibody, could be insulin gene, okay? And there is a lot of genetic diseases in the world. Think about albinism. This is an illness that there is a defect in the gene that produces melanin. Okay, and you also have ocular albinism, which is the same thing, but these people are very often blind because they don't have the pigment melanin in the eye, and then you cannot see. So this is like a black person with ocular albinism. It's possible to have both albinisms at the same time. There is another condition which is called heterochromia. This is not really a condition because it's even beautiful. This is very rare because usually your eyes, they're controlled by the same genes. And if you have heterochromia, this means that one eye is according to your father and the other one is according to your mother, and you have two different colors, but you already need that your two parents have two different colors. So I met only twice a person in my life who has heterochromia, and I think it's really nice, but it's very, very rare. But basically it's not really a condition. It just implies that the melanin of your eyes, it's coded by two different genes, which is very rare. And DNA mutations, you all know, we're going to see that, especially next semester, that DNA a mutation is, for instance, an A changing in a T or a G changing in a C or in an A or whatever. One mutation can be enough to create cancer or serious genetic illnesses. And that's why the DNA replication, it's 99.9999% perfect. There is almost no mistakes happening there. But when a mistake occurs, it's dangerous because it can cause illnesses. So this, we have also seen it, guys. This is basically the difference between DNA and RNA. So here you see a DNA strand, which is double, and it's in Spanish, but we have the bases, cytosine, guanine, adenine, and thymine. Okay, these are the bases of DNA. The basis is when we refer only to the letters, nucleotides when we refer to the whole molecule. Okay, RNA is only single-stranded and it has different bases. I mean, they have all the same bases except for uracil. It's a different base in RNA. Okay, that's what we also have seen. Here you can see a little bit more about the features of each one of them. I'm not going over that again. This one, yes, because it's important. Remember the process. What is gene expression? It's a DNA, okay, becomes basically a protein. And I'm going to explain that, how that works. So I like apple pie, okay? So in order to make an apple pie, you could go to the library and take a cookbook, a cooking book, okay? But of course, the cooking book, it has the DNA, this library, you have to leave the books there and the book cannot leave the library. So it's the DNA in the nucleus. So the only thing you can do is make a copy. So what I will do is I take a copy of my recipe that I want of the big cooking book, and I will only take apple pie recipe. Okay, that's my messenger RNA because that's the copy I did. Okay, so DNA is the book, RNA is the page that I want. Okay, and this is called transcription. The recipe, I take it with me and I go home. Okay, my home is endoplasmatic reticulum and the ribosome is my mom because she helps me with apple pie. So what is translation is basically I read my recipe of my apple pie and I'm gonna translate my recipe in a real pie. But of course I need ingredients, okay? And these ingredients, they come from the transfer RNA because it's the proteins, the amino acids, sorry. So it's my mom bringing the amino acids for me to make the pie, which is the protein, okay? Everybody gets it?
I have one question.
Yes, go ahead.
For transcription, does the RNA copy the template or the non-template strand?
It copies the look, if the template strand is what we call the coding strand. So usually the coding strand has the gene, okay? So if you want to copy that certain gene, you have to copy the non-template strand in order to have the same fragment, right?
Yes.
Yes.
Okay.
So basically the coding strand has the gene. So you're not copying the gene, you're copying the template strand of the gene in order to have the same sequence. Okay? Good.
Okay.
In fact, because that's in fact the thing that for some students is not really clear. Okay, and then here you see the same image again. Here is the gene. I copy the template strand of the gene that I want. I get exactly the same piece of messenger RNA. Then is the DNA with the only difference that T is U. Then the messenger RNA goes to the ribosome. The ribosome, which is ribosomal RNA, travels on top on the messenger RNA. Then the transfer RNA brings in the ingredients and then the protein. You see here the tail becoming longer and longer. Okay, it's basically in theory it's very simple, the process. Okay, now to finish the class of today, we're going to talk a little bit about the Human Genome Project. I'm not going into much detail. The only thing I want to tell you about the Human Genome Project, it's a project that got a lot of recognition, and it was finished, I believe, in the year 2000. And it's basically that in the year 2000, when the Human Genome Project was finished, is that they sequenced the whole DNA of a human. So basically we have the whole DNA now, the letters, the whole strand. We know now each letter of the human DNA. Of course, this is different in different humans, but this was a very important moment in science because it gave us a very deep understanding in genetic diseases, in how the genome works, and in which diseases are related with the genes, let's say. And one of the most important ones, which is causing the most deaths in the world, is cancer. You know, cancer is a genetic disease, but we will see that in the future in much more detail. Here is again the history of how they discovered the DNA. We did that yesterday. You remember these scientists, how little by little they identified the molecule DNA and they ruled out it was not RNA, it was not protein, and how finally Watson and Crick, they discovered how the molecule really looked like, that it was a double helix, okay? This is the video we saw yesterday, and this is the video I wanted to show you tomorrow, okay? And here, to finish the class of today, I want to check some techniques in biotechnology. You just have to know what those terms are, okay? We don't have the time to go in a lot of detail, but I go over them for you to understand what it's all about. If you have heard about recombinant DNA, recombinant DNA is basically when the DNA is not only of one organism, okay? So it's a hybrid. So in this case, they made once an organism, an animal that was half goat and half sheep. And we call that recombinant DNA because we recombined the original DNA with other DNA to make a new creature. Okay, this creates a lot of ethical issues, of course, because recombinant DNA, the thing is, there is many mistakes in it. We are still not as really good at it, and we're making basically Frankenstein organisms. And there you have to ask yourself, what are we playing with? You know, so it's very experimental, yeah, but we do it more and more nowadays. Hybridization refers to the fact of DNA combining with another strand of DNA. So that's what hybridization is, you know. Hybridization is just one strand combining with another strand, okay? Here in this example, they show DNA of one species and another species. You can open it up and you can connect it together again, but you see some excess here. It's where they don't match, but the molecule still combines. And that's the problem with this sheep-goat thing, because these animals, they get genetic illnesses because there's a lot of mistakes in the DNA, okay? PCR, you have heard of it, because I suppose some of you, they did already COVID PCR test, okay? So basically, what is a PCR? It's a revolutionary technique to amplify DNA. It amplifies DNA in an exponential way. So here you see one piece of DNA, and basically what it does, it opens the DNA and it makes a copy. It opens the copies and it makes 4 copies. So you go from 2 to 4, from 4 to 8, from 8 to 16, so 16 to 32. And it's a method to make from one piece of DNA a lot of DNA. So it's really nice to make copies and copies of something you're interested in. It's also interesting for testing for coronavirus, because there is very little DNA of the virus in you. So if you have like one little piece of DNA, by copying it, you get like a lot, and you have absolute certainty if you're infected with coronavirus or not. So this technique has a lot of applications, and the people that invented the technique, they even won the Nobel Prize for it. Electrophoresis is another technique that we use a lot in genetics and biotechnology. And DNA of different lengths, and DNA has a special pattern, okay? Each piece of DNA has a certain barcode, like you can see here. If you do it with gel electrophoresis, a piece of DNA looks like this. So let's assume that this is used in forensics. And what you see here in the middle is the DNA they found on the site where the crime happened, okay? And here you have suspect 1, 2, 3, 4, 5, 6. 1, 2, 3, 4, 5, 6. So if you find this piece of DNA on the crime scene, so who of the suspects is in fact the guilty one?
3.
Number 3.
Exactly 3, because it has exactly the same pattern. And that's why DNA is, without any doubt, it gives us 100% certainty of who did the crime. And it's been used in solving a lot of crimes because of the uniqueness of it, because every person would have a different pattern. And with this, it's 100% proof that if you left your DNA on the crime scene and they take your DNA and it matches, of course, you're the guilty one. Okay, and then clonation, this happened even before you were born. In 1996 and 1997, they cloned Dolly, which was the first sheep, the first animal that was cloned. What they did is they took an ovolo of a certain sheep, and they would fill it with DNA from another sheep, and then they would plant it in the uterus, and an exact copy of the sheep, of the donor sheep of the DNA would be born again. The only problem with the sheep Dolly is that the sheep died because there were like genetic secondary effects which caused the animal to develop cancer and die premature. Okay, so that's basically it, what I wanted to tell today. I'm going to share the video that I wanted to show you the process of transcription and translation. I'm going to find the video and I share it with you in the WhatsApp group, because in fact the lesson of today is finished, and tomorrow we have an activity and we have the quiz. I will also not have the time tomorrow to see this, but I hope everything is clear. Any questions? No? I'm sorry, I know it was a lot of information, but it's the only way to do it like this is explain it step by step. I hope I made myself clear. and let's see how you do tomorrow in that little quiz, okay? And then tomorrow we have also an activity, and for the rest, I hope you enjoy certain talks of Changemaker Day, and we see each other tomorrow, okay?
Thank you, teacher. Bye.
Thank you.
Thank you.
Thank you, Mr.
Take care, bye-bye.
Ismail.